कैसे हैं आप सब लोग? आई होप आप सब लोग बिल्कुल ठीक हैं। सो वेलकम टू वीडियोलॉजी बाय अत्याशा। बिस्मिल्लाहिर्रहमानिर्रहीम। हम चलते हैं अपनी वीडियो की तरफ। तो इस वीडियो से हमने टॉपिक स्टार्ट करने हैं इस वीक के भी, और जितने भी टॉपिक्स आते हैं बायो इन्फॉर्मेटिक्स के, क्योंकि इसका क्विज़ है हमारा। आप लोग को मैंने पिछली वीडियो में क्विज़ की डेट्स वगैरह भी बताई थी, और इसका क्विज़ स्टार्ट हो रहा है जनाब कल ही। आज 29 है तो कल 30 है, तो कल ठीक है, कल स्टार्ट होगा 2 दिसंबर तक जाएगा। इसलिए मैं चाहती हूँ कि मैं आज ही इसके टॉपिक्स भी कर लूँ और इसके क्विज़ की भी तैयारी कर लूँ। ठीक है, आई विल ट्राई माई बेस्ट। सो चलते हैं यहाँ पर, और इसकी प्री असेसमेंट ले लेते हैं। सबसे पहले सही है, प्री असेसमेंट ले लेते हैं। RNA sequences are determined by protein templates as well as DNA. अच्छा, प्रोटीन टेम्पलेट्स से DNA और RNA दोनों के टेम्पलेट्स जो है वो सीक्वेंस किए जा सकते हैं। मेरे ख्याल में ट्रू है। सही, आगे चलते हैं। तो ये फॉल्स था, ठीक है, कोई मसले वाली बात नहीं। हम आगे चलते हैं, हम अपनी वीडियो में आ जाते हैं। वीडियो को अच्छे से सुन लेते हैं।
Dear students, in this module, we're going to discuss the various types of nucleotides that are there in the DNA and the RNAs. As you would know by now, that the DNA is comprising of 4 different nucleotides. There are multiple copies of these nucleotides within a single DNA molecule. In case of the RNA, it is also comprised of 4 different nucleotides. And an RNA molecule can have multiple copies of these nucleotides as well. In this figure, you can see that in case of the DNA, you have thymine, adenine, cytosine, and guanine, and they are coupled by sugar and phosphate backbones. To discuss this backbone that is holding the entire nucleotide bases together here, as well as on the other strand, let me briefly mention here that the sugars that are there are different in case of RNA and DNA. So you essentially have a sugar, a phosphate, a sugar, a phosphate, and so on, which is actually shown here as well. Phosphate is shown by the P, while the sugar is shown here. So this backbone differs between the DNA and the RNA by the sugars. So in case of the DNA, the sugars are 2-deoxyribose, while in case of the RNA, it's just the simple ribose. So remember this difference in the sugars between the DNA and the RNA. Their coupling to the phosphate stays the same. So it is a phosphate on top of a sugar on top of another phosphate on top of another sugar. The nucleotide bases are attached to the sugars. So there are these 5 different nucleotides if you look at the DNA and the RNA together. So you have adenine and guanine drawn here. So these are termed as the purines due to their structure. And then you have the cytosine, uracil, and thymine, which are the pyrimidines. So the difference here is in the structure. The purines have 2 rings, while the pyrimidines have a single one. Moreover, in case of the DNA, you only have the cytosine, the adenine, guanine, and thymine, while in case of RNA, you have adenine, guanine, uracil, and cytosine. So in this way, the RNA and DNA differ between uracil and thymine. Now, how do these phosphates, sugars, and nitrogenous bases, or these nucleotides, they come together? So if you see here carefully, the phosphate group is shown here, the sugar is shown here. In case of RNA, you will have another OH here. In case of the DNA, you will only have a hydrogen here. So on top of this, the nucleotide base comes and attaches itself. So in this way, the phosphate sugar chain continues, and newer nucleotides, they come and attach themselves, and the DNA or the RNA molecule is constructed. So in conclusion, the DNA gives rise to the RNA, and the DNA has 4 nucleotide bases, that is A, T, C and G, while the RNA has 4 bases A, T, U and G. More so, DNA is double-stranded, RNA is single-stranded, and the RNA molecule is used towards production of the proteins.
सो फ्रेंड्स, हमारी वीडियो हो गई है कंप्लीट। अब हम चलते हैं अपनी अगले स्टेप की तरफ। अगला स्टेप हमारा है रीडिंग का। रीडिंग में चल लेते हैं हम। इसकी सबसे पहले पीपीटीज डाउनलोड कर लेते हैं। आई होप सो जल्दी से पीपीटीज हमारी डाउनलोड हो जाए। यस, और पहले सबसे यहाँ से पढ़ लेते हैं, ठीक है। इंट्रोडक्शन। अ न्यूक्लियोटाइड इज द बेसिक बिल्डिंग ब्लॉक ऑफ न्यूक्लिक एसिड्स। RNA and DNA are polymers made of long chains of nucleotides. Our nucleotide consists of a sugar molecule, either ribose in RNA or deoxyribose in DNA, attached to a phosphate group and a nitrogen containing base. Types of nitrogenous bases. There are 5 types of nitrogenous bases, और वो क्या हैं? एडिनाइन, साइटोसाइन, गुआनाइन, थाइमाइन, यूरेसिल। स्ट्रक्चर ऑफ DNA एंड RNA। अब इनका स्ट्रक्चर, DNA molecule although is double stranded and RNA is single stranded, but there is difference in sugar composition and in one nitrogenous base. In DNA, thymine present while in RNA uracil is present. RNA has ribose sugar and DNA has deoxyribose sugar. सही हो गया? ओके। उसके बाद एडिनाइन एंड गुआनिन कलेक्टिवली कॉल्ड प्यूराइन्स, साइटोसिन, यूरेसिल एंड थाइमाइन आर कॉल्ड पाइरिमिडाइन्स। सही है। और इसके अलावा व्हेन फॉस्फेट नाइट्रोजन बेस एंड sugar comes together, if there is OH then the molecule is RNA, and if there is H in sugar then the molecule is DNA. I'm sorry. चलते हैं आगे. So in conclusion, we have DNA molecule make RNA and RNA make the protein, and DNA differ from RNA in nature due to sugar and nitrogenous base. DNA just codes the information for proteins, but RNA helps in making proteins. सही हो गया, बिल्कुल ठीक. अब हम चलते हैं अपनी PPTs की तरफ. हम बिल्कुल भी वेस्ट नहीं करेंगे टाइम को और फ़ौरन से PPT की तरफ चल लेंगे. So friends, let's go towards our PPTs. These are about to come. So almost there. Okay. अब हम चलते हैं इसकी तरफ और इसको करते हैं बड़ा. न्यूक्लियोटाइड्स सीक्वेंस एनालिसिस, और इसके अंदर है न्यूक्लियोटाइड्स और उसके बैकग्राउंड. डीएनए एंड आरएनए मॉलिक्यूल्स आर built up from 4 types of molecules each. These molecules are called nucleotides. These are adenine, cytosine, thymine, uracil and guanine. ठीक है. उसके बाद शुगर्स होती है. डीएनए के अंदर 2 डीऑक्सीराइबोस होती है, आरएनए के अंदर राइबोस शुगर होती है. और बैकबोन कैसे होती है? पहले शुगर होती है, फिर फॉस्फेटिक ग्रुप होता है, फिर उसके ऊपर शुगर होती है, फिर उसके ऊपर फॉस्फेट ग्रुप होता है. और हर एक जो शुगर है उसके साथ में उसकी बेसिस अटैच होती हैं. न्यूक्लियोटाइड्स के अटैच होते हैं. कौन कौन से होते हैं? थाइमाइन, आर्जिनाइन, साइटोसाइन और गुआनिन. ठीक है. इसके बाद हम चलते हैं आगे. स्ट्रक्चर ऑफ एडेनाइन, साइटोसाइन, थाइमाइन, यूरेसिल एंड गुआनिन. ये प्यूराइंस में एडेनाइन गुआनिन आ जाती है, पाइरिमिडाइन में साइटोसिन, यूरेसिल एंड थाइमाइन आ जाती है. डन हो गया। उसके बाद फॉस्फेट शुगर एंड नाइट्रोजन बेसिस कम टुगेदर। अब यहाँ पर ये फॉस्फेट ग्रुप है, ये शुगर है, शुगर के साथ अटैच हुई नाइट्रोजन बेस। अब इसके अंदर ये जो आर ग्रुप है शुगर के अंदर, उसमें अगर OH होगा तो वो RNA में होगा, और अगर हाइड्रोजन होगा आर की जगह पर तो वो DNA में होगा। सही है, कंक्लूजन में आ गया जनाब। DNA gives rise to RNA. RNA has adenine, thymine, cytosine and guanine, while RNA has adenine, uracil, guanine and cytosine. DNA is double stranded while RNA is single stranded. RNA helps produce proteins. ठीक है, आई होप आप लोग को मेरी आवाज़ सही आ रही होगी। सो अब चलते हैं हम लोग अपने इस टॉपिक के लास्ट स्टेप की तरफ। फिर उसके बाद ये टॉपिक जो है ना बिल्कुल भी कंप्लीट हो जाएगा। ठीक है, सो लेट्स मूव ऑन। तो इसका कुछ ले लेते हैं पोस्ट असेसमेंट का। हाँ जी, सो हियर वी हैव द कोडोन इज अ डैश डुप्लेट सिंगलेट ट्रिपलेट। इट इज अ ट्रिपलेट। सो इसको करते हैं सेव। आगे चले। विच association between complementary bases would require the most energy to break, has the highest bond dissociation energy? Adenine or uracil, adenine, thymine, cytosine और guanine। हाँ, cytosine और guanine आई गेस। सही है, इसको करते हैं सेव। वो कर करेक्ट। थैंक्स लॉर्ड। अब चलते हैं आगे। हमारा अगला टॉपिक जो है उसके बाद चल लेते हैं। सॉरी, ठीक है। टॉपिक नंबर 36 जेनेटिक कोड। ठीक है, अब इसके क्विज ले लेते हैं सबसे पहले। तो री असेसमेंट। व्हाट इज द रिलेशनशिप अमंग DNA, अ जीन एंड अ क्रोमोसोम? देखते हैं क्या रिलेशनशिप है। क्रोमोसोम कंटेन्स hundreds of genes which are composed of proteins. A gene contains hundreds of chromosomes which are composed of protein. A chromosome contains hundreds of genes which are composed of DNA. A gene is composed of DNA, but there is no relationship. अच्छा, I think तो ये बेहतर है। Let's see, is correct. Yes. अब इसको कर देते हैं क्रॉस। चलते अपने वीडियो की तरफ, इसको अच्छे सुन लेते हैं।
Now let's talk about the genetic code. This is basically the code that translates the nucleotide information into the protein or amino acid information. There are 3 messenger RNA nucleotides that form a codon. These 3 nucleotides basically imply that which amino acid will be placed in a particular protein at that region. This genetic code is complementary and anti-parallel to corresponding DNA. Genetic code is basically the sequence of nucleotides of messenger RNA. The 3 nucleotides at a time, 3 adjacent nucleotides of the messenger RNA, they basically specify a particular amino acid. That's basically what we are talking about. Genetic code relates codons to their specific amino acids in proteins. The important features of genetic code is that it is universal and redundant but not ambiguous. So what do I mean by that? Universal is that pretty much all organisms, they use the same genetic code. There are a few minor exceptions, but pretty much the genetic code is universal. So if we take a messenger RNA from a human and put it in a bacteria will make the same protein a human cell would, because the genetic code is almost universal. Redundant meaning that more than one codons specify one particular amino acid. On the screen you can see, for example, for leucine we have 6 codons. All these codons specify leucine. Wherever these codons appear in messenger RNA, leucine will be placed in the resulting protein. It is not ambiguous, meaning that these codons will only result in leucine. It does not mean, by ambiguous we mean sometimes a codon will specify leucine, sometimes it will specify serine, sometimes it will specify proline. That doesn't happen. It is redundant but not ambiguous. It is always specific. There's also start and stop codons. Here I have placed the whole genetic code on the screen. So you can see we have first letters here, the second letters here, and third letters of the genetic code. It's a 3-letter code, and the combinations are here. Here I would like to point out that AUG is codes for thionine, and this is also the start code. This is a codon which tells where to start making the protein from on a messenger RNA. We'll talk more about that. There are also 3 stop codons. These 3 stop codons, whenever they appear in messenger RNA, they basically tell the ribosomes to stop making protein when they hit this stop codon. So how was genetic code deciphered? How did people know which 3 nucleotides specify which particular amino acid in a protein? There are simple experiments initially that basically people were able to synthetically made polynucleotides of the same nucleotide. For example, poly-U chain, uracil, as you know, is one of the nucleotides in the messenger RNA. When they made this, artificially made this messenger RNA and placed in cell extract with the machinery which could translate it, convert it into protein, all the amino acids in the protein were phenylalanine. So basically meaning that 3 U's basically code for phenylalanine. Similarly, 3 A's code for lysine, 3 C's code for proline. So gradually people were able to come up with complete the genetic code, which 3 nucleotide sequence or the codon specifies which amino acid. Now we will talk about the adapter, because adapter is the molecule that interacts with the codons, and as we have mentioned earlier, adapter has to have 3 functions. It has to carry an amino acid. On one domain of this adapter should carry amino acid. It should be able to associate with messenger RNA, because it has to interact with the codons. It has to be able to associate with messenger RNA. It should be able to bind ribosomes, because these organelles are the ones responsible for synthesizing proteins. So tRNA is the adapter. It is, as the name implies, it is an RNA molecule, but we put a small t here, meaning that it's a transfer RNA. So we have talked about one type of RNA was messenger RNA, which is carrying the message from the nucleus to the cytoplasm. This is the transfer RNA. It is about 75 to 80 nucleotides long, and it performs all these functions. So nucleotides, this tRNA, all these, we have several tRNAs, each carrying a specific amino acid and can interact with a specific codon. It has a sort of a little complex 3-dimensional structure, which we'll look at. That structure is due to the internal hydrogen bonding. I'll show you that. Conformation, the 3-dimensional shape or placement of atoms in 3-dimensional space of this molecule allow it to bind the ribosomes. The 3' end of the tRNA, we know exactly what 3' end is, basically implying the 3' hydroxyl of the last nucleotide of this molecule is the place where amino acids are bound. Midpoint of this molecule tRNA has an anticodon. Anticodon is basically the part of this molecule that interacts with the codon present on the messenger RNA. So you see the beauty in it. This molecule has nucleotides. These nucleotides have a sequence which can bind specific codon on the messenger RNA, and on other tip it is carrying an amino acid. So this is basically the functions of the adapter or the transfer RNA. I have to again point out that the interaction of codon and anticodons is antiparallel. All nucleotide interactions are antiparallel for our purposes. Here's a 3-dimensional structure. Here you can see the internal 3 hydrogen bonding which is forming between the nucleotides. We know that A's can have hydrogen bond with the C's, A's can have hydrogen bonds with U's, and G's can have hydrogen bonds with C's. So this basically results in formation of this structure, three-dimensional structure, and the 3' end is where the 3' hydroxyl is. This is the part where amino acid will be bound. And just so that you know, the abbreviation we will use in later slides for tRNA is this. So we have seen the structure of tRNA, and next we will talk about what is the role of this tRNA in protein synthesis, and first of all how it gets to carry the amino acid. We'll look at that later.
सो फ्रेंड्स, हमारी हो गई है कंप्लीट, हमारी वीडियो कंप्लीट हो गई है. अब हम चलते हैं अपनी रीडिंग की तरफ. इसको डाउनलोड कर लेते हैं और इसको रीड आउट कर लेते हैं. ठीक है. जेनेटिक कोड. हाउ डू ट्रांसक्रिप्शन एंड ट्रांसलेशन प्रोड्यूस स्पेसिफिक एंड फंक्शनल प्रोटीन प्रोडक्ट्स. ठीक है. These processes require a genetic code that relates genes DNA to mRNA and mRNA to the amino acids of proteins. The genetic code specifies which amino acids will be used to build a protein. You can think of the genetic information in an mRNA molecule as a series of sequential non-overlapping 3-letter words. Each sequence of 3 nucleotide bases the 3 letters. ठीक है. Along the chain specifies a particular amino acid. Each 3-letter word is called a codon. Each codon is complementary to the corresponding triplet in the DNA molecule from which it was transcribed. Thus, the genetic code is the means of relating codons to their specific amino acids. The complete genetic code is shown in the figure, and इसके बाद notice that there are many more codons than there are different amino acids in proteins. Combinations of the four available letters, the bases, give 64 or 43 different 3-letter codons, yet these codons determine only 20 amino acids. AUG, which codes for methionine, is also the start codon, the initiation signal for translation. 3 of the codons, UAA, UAG and UGA, are stop codons or termination signals for translation. ठीक है. When the translation machinery reaches one of these codons, translation stops and the polypeptide is released from the translation complex. ठीक है. इसके बाद क्या होता है, ये सारा का सारा टेबल है. ठीक है. ये हमने याद कर लेना है इंशाल्लाह. टेबल सही है, वो बिल्कुल भी वो नहीं है, ये मुश्किल, बहुत आसान है. ठीक है. प्रिपेयर अ बैक्टीरियल एक्सट्रैक्ट containing all the components needed to make protein except mRNA. ठीक है. Add an artificial mRNA containing only one repeating base. The polypeptide produced contains a single amino acid. तो 3U जो है वो ट्रिपल U रिप्रेजेंट करेंगे फिनाइल एलेनाइन. ठीक है. फिनाइल एलेनाइन को. उसके बाद फिर ट्रिपल A करेंगे रिप्रेजेंट लाइसाइन को, और ट्रिपल C करेंगे प्रोलाइन को रिप्रेजेंट. ठीक है. ओके. द जेनेटिक कोड नायरनबर्ग एंड मैथी used a test tube protein synthesis system to determine the amino acids specified by synthetic mRNAs of known codon composition. ठीक हो गया जनाब. अब हम पढ़ लेंगे इसकी PPTs. So let's go on. Genetic code. ठीक है. 3 mRNA nucleotides form a codon which is complementary and antiparallel to corresponding DNA. Genetic code relates codons to their specific amino acid. Universal and redundant but not ambiguous. Some exceptions start and stop. ठीक है. ये भी सेम होगी, बात सेम हो गई. ठीक है. tRNA translates nucleotide language into amino acid language, carries amino acid associated with the mRNA and binds राइबोसोम्स, इसकी एबिलिटी होनी चाहिए राइबोसोम के साथ बाइंड करने की, ठीक है, एमआरएनए के साथ एसोसिएट करना चाहिए इसको और कैरी करना चाहिए अमाइनो एसिड को, ठीक है। उसके बाद हमारे पास में टीआरएनए हैज 75 to 80 न्यूक्लियोटाइड्स विद इंटरनल हाइड्रोजन बॉन्डिंग, ठीक है। कन्फर्मेशन ऑफ टीआरएनए अलाउस इट टू बाइंड राइबोसोम्स। 3 प्राइमर एंड ऑफ टीआरएनए CCA is attached to amino acid and midpoint has anticodon antiparallel, सही। उसके बाद फिर है हमारे पास में ये सारा का सारा दिस कंप्यूटर जनरेटेड स्पेस फिलिंग रिप्रेजेंटेशन शोज द 3-dimensional structure of tRNA, ठीक है। दिस आइकन फॉर tRNA विल बी यूज इन द फिगर्स दैट फॉलो, सही है। इसकी जगह पर ये यूज होगा, ठीक है। उसके बाद दिस 3-dimensional representation of the internal regions of the base pairing, ठीक है, सही। ये वाले बेस पेयरिंग के रीजन है वो एम्फसाइज कर रहे हैं इसमें, ठीक है। द एंटीकोडॉन कंपोज्ड ऑफ द 3 bases that interact with mRNA or from the amino acid attachment site, ठीक है। उसके बाद दिस flattened cloverleaf model emphasizes base pairing between complementary nucleotides, ये 3 प्राइमर एंड है, ये सारा क्या है टीआरएनए ट्रांसफर आरएनए, ठीक है, ओके। सो ये ट्रांसफर आरएनए है, इसको कर लेते हैं क्रॉस। यहाँ तक हमारा ये टॉपिक जो है वो ऑलमोस्ट कंप्लीट है, बस इसकी प्रीवियस पोस्ट असेसमेंट रहती है और उसको भी हम कर लेते हैं, ठीक हो गया। क्विज ले लेते हैं। डैश इज द process by which 2 identical copies of DNA are made from an original molecule of DNA, that is duplication. 2 identical copies of DNA are, हाँ ये है duplication. UAG, UGA and UAA are all stop codons. Yes, true. कर देते हैं सेव, ये हो सकता है फिर एप्लीकेशन, ठीक है। सो इसको कोई मसले वाली बात नहीं, हम इसको कर देते हैं क्रॉस। हम चलते हैं आगे, अगला टॉपिक हमारा है टॉपिक नंबर 37 ट्रांसक्रिप्शन 1, ठीक है। प्री असेसमेंट इसकी ले लेते हैं। टेक क्विज। UAG, UGA एंड UAA आर ऑल स्टॉप कोडॉन्स, ट्रू। इसको सेव कर देते हैं। वेल इसको करते हैं हम क्रॉस, इसको दोबारा से ले लेते हैं। अब इस एलएमएस की वर्किंग बहुत ख़राब हो रही है आजकल, बस अल्लाह करे हमारा कुछ अच्छा सा हो जाए, उसमें प्रॉब्लम ना आ जाए कुछ में। इसको रीटेक करें, ये असेसमेंट से हम रीटेक कर सकते हैं कि कुछ रीटेक नहीं कर सकते। स्टार्ट कोड ऑन इज़ AUG है मेरे ख्याल में, तो नहीं UGA रीटेक करते हैं और इसको तो सेव है। स्टार्ट कोड ऑन इज AUG, वही है AUG था। करते हैं क्रॉस, उसके बाद हमारा वीडियो आगे, वीडियो को अच्छे से सुन लेंगे।
Now we look at the process by which DNA makes a copy of itself, but this copy is not a DNA molecule, it's an RNA molecule. This process is called transcription, transcribing the information which is contained in DNA. Transcription initiation begins at the promoter. Promoter is a sequence of nucleotides. This promoter has specific functions. One of that function is that it can bind RNA polymerase. So RNA polymerase is the molecule that will of course make the RNA, but it binds specific region of DNA which has the promoter. Next to promoter are genes, which will be of course their RNA will be used to make proteins. Promoter tells specific information to the RNA polymerase. It tells the RNA polymerase where to start the transcription, where to start reading from, where to start manufacturing RNA from. It also tells RNA which DNA strand to read. As you know that DNA is a double helix, there are two strands of course in the DNA. One of them, the template strand, is used to make RNA, and the strand which is complementary to this template strand is not used to make RNA. So we can call complementary strand a non-template strand also. So this is one of the information that promoter provides to RNA polymerase. It also gives RNA direction where to take a start, that it should start transcribing or making RNA. Part of the promotion is the initiation site where the transcription will start, meaning that's the part of DNA which will get converted into messenger RNA is also part of the promoter. Here, once the RNA polymerase has landed on the promoter, it starts putting in the nucleotides. Now these are the RNA nucleotides, not the DNA nucleotides. RNA polymerase cannot use the DNA nucleotides. RNA polymerase moves from 3' direction to 5' direction. This is the direction in which RNA polymerase is moving. As I have mentioned before, all polynucleotide synthesis, whether it is DNA or RNA, it only can be extended or can be made in 5' to 3' hydroxyl. Remember, we have talked about 3' hydroxyl, but RNA polymerase is basically moving in direction 3' to 5' direction. Synthesis of RNA is in 5' to 3' direction. Here I would like to mention that no primer is needed. When we talked about DNA replication, I told you DNA polymerase cannot start the replication of DNA. It needs a primer, and RNA inside the cell, RNA, a special type of RNA polymerase, makes that primer to start the DNA replication process. The RNA polymerase unwinds 20 base pairs at a time, and as it is moving along, it is making the RNA transcript. The transcript is anti-parallel to DNA template strand. We know exactly what that means. We have talked about that in detail. Another difference between making RNA and making DNA is, we have talked about this, that DNA errors cannot be tolerated. If there is a change in DNA, it is the genetic information, the master blueprint of the cell. It is conveyed from one generation to the next generation, and that can have catastrophic results. But RNA is not a hereditary molecule for at least the eukaryotic cells. There is no proofreading ability in the RNA polymerase. It makes the error rate is about 1 in 10,000 to 100,000. So this RNA polymerase makes mistakes, but that's okay, because RNA is short. It is made and it is degraded fairly rapidly, but it has enough time to make the proteins, but it does not last very long in the cell. So once the RNA polymerase has made the RNA, it encounters the termination site. The termination site is also a sequence of nucleotides that tell RNA polymerase to stop the transcription process. Here I would like to mention difference between prokaryotes and eukaryotes. In prokaryotes, the DNA is not enclosed in the nucleus. It is out there in cytoplasm, in the nucleoid region. So as the RNA is being made, the translation process also starts, meaning the RNA is being simultaneously, concurrently made, also being converted into a protein molecule. In eukaryotes, the first messenger RNA that is made is called the pre-messenger RNA, and it goes through modifications. We'll talk about those modifications later, but the first RNA made by eukaryotes in the nucleus is the pre-mRNA. So here there is an animation, you can see it on the screen. The RNA polymerase, it binds the promoter, it separates about 20 base pairs at a time. It starts incorporating nucleotides. These nucleotides are complementary to the template strand, to the nucleotides of the template strand, and this transcript is also anti-parallel. We know what that means. This process keeps continuing till the RNA polymerase hits the terminator, or the site where there is information for RNA polymerase to stop the transcription process. When RNA polymerase hits that site, it disengages and the messenger RNA is released. Now this messenger RNA is ready for, if it is a prokaryote, it gets start, the translation process starts right away. If it's a eukaryote, this pre-messenger RNA that we just made on the screen will first go through certain processes before it can be used to make protein molecules.
सो फ्रेंड्स, फाइनली हमारी वीडियो हो गई है कंप्लीट. हम चलते हैं अपने रीडिंग की तरफ. फटाफट से रीडिंग कर लेते हैं, क्योंकि इसको डाउनलोड कर लेते हैं. ट्रांसक्रिप्शन था. Formation of a specific RNA from a specific DNA requires several components. A DNA template for complementary base pairing, the appropriate ribonucleotide, triphosphate, ATP, GTP, CTP, and UTP to act as substrate and enzyme. RNA polymerase. सही. Within each gene, only one of the two strands of DNA, the template strand, is transcribed. The other complementary DNA strand, referred to as the non-template strand, remains untranscribed for different genes in the same DNA molecule. In the same DNA molecule, different strands may be transcribed. That is the strand that is the non-template strand in one gene may be the template strand in another gene. ठीक है, सही है. और ये सारे का सारे डायग्राम सर ने पहले ही समझा दिए, इसमें वही बात है. तो अब चलते हैं हम अपने PPTs की तरफ. उनमें क्या है, सही है. फिर उसमें यहाँ पर आ गया ट्रांसक्रिप्शन. ट्रांसक्रिप्शन में हमारे पास इनिशिएशन बिगिंस एंड प्रोमोटर टाइटली बाइंड्स RNA पॉलीमरेज. ठीक है. यहाँ पर आ गया वे टू स्टार्ट ट्रांसक्रिप्शन, विच स्ट्रैंड टू रीड. ये किसके हैं, ये अडाप्टर उसके, ये इसके प्रोमोटर के जो है ना फंक्शन है ये. सही है. वे टू स्टार्ट ट्रांसक्रिप्शन और विच स्ट्रैंड टू रीड द डायरेक्शन टू मेक टू टेक स्टार्ट फ्रॉम. पार्ट ऑफ प्रोमोटर इज इनिशिएशन साइट एंड ट्रांसक्रिप्शन स्टार्ट्स हियर. उसके बाद RNA polymerase moves 3 to 5 primer. Synthesis is done from 5 to 3 primer. Primer unwinds 20 base pairs at a time. ठीक है. ट्रांसक्रिप्ट एंटी पैरेलल टू DNA template strand. नो प्रूफ रीडिंग एरर, नो प्रूफ रीडिंग बिल्कुल भी. एक बार जो जीन बन जाती है, जो कोड हो जाता है, वो दोबारा से रीड आउट नहीं किया जाता, गो थ्रू नहीं किया जाता कि सही है कि नहीं. जैसा बन जाता है बस वो ही चलता है. और ऑफ कोर्स अल्लाह ताला का करिश्मा, अल्लाह ताला के जलवे हैं, ये उसकी शान है, तो उसकी चीज़ों में गलती कैसे आ सकती है. सो यहाँ पर फिर एरर रेट आप लोग देख लें 1% है. 1 एरर रेट है 1% इन 10,000 to 100,000 जीन्स. इससे सारे कोड्स में, इतनी सारी चीज़ में 1% चांस होता है उसका मिस्टेक का, और ये भी अल्लाह ताला की अपनी मर्जी होती है. सही है. अल्हम्दुलिल्लाह, अल्लाह का शुक्र. चले जी, अब चलते हैं हम आगे. ट्रांसलेशन स्टार्ट्स बिफोर टर्मिनेशन इन प्रोकैरियोट्स. ठीक है. प्री mRNA फर्स्ट प्रोडक्ट ऑफ ट्रांसक्रिप्शन इन यूकैरियोट्स. हाँ, सर ने बोला ना कि जैसे जुवेनाइल RNA polymerase reaches the termination site, that RNA transcript is set free from the template. हाँ ठीक है ना, हमें सर ने एक एनिमेशन भी दिखाई है, उसमें भी ऐसा ही हो रहा है. सही है. तो प्रोमोटर में स्टार्ट होती है, टर्मिनेटर में एंड हो जाती है ट्रांसलेशन. ठीक है. ट्रांसक्रिप्शन ठीक है, जिसमें RNA कम्प्लीमेंट्री स्ट्रैंड से RNA बन रहा है, DNA के कम्प्लीमेंट्री स्ट्रैंड से RNA बन रहा होता है. वो सारे के सारे कोड जो है ना उसके ऊपर लग रहे होते हैं एक स्ट्रैंड के ऊपर से, तो उसके बाद में RNA बन जाता है. प्रोकैरियोट्स में वहीं से ही ट्रांसलेशन स्टार्ट हो जाती है, लेकिन यूकैरियोट्स में जो है वो कुछ प्रोसेसेस से वो गुजरता है. mRNA के बाद वो tRNA बनता है, tRNA के बाद ही वो ट्रांसलेशन स्टार्ट करता है. और अब इसको कर देते हैं हम क्रॉस. हमारा टॉपिक यहाँ पर ऑलमोस्ट एंड है. हम प्रोसेस असेसमेंट ले लेते हैं. ठीक है. तो इन यूकैरियोट्स इन ऑर्डर टू इनिशिएट ट्रांसक्रिप्शन RNA polymerase must be present. None of these RNA strand must be present. Promoter sequence must be present. हाँ आगे चलते हैं. स्टार्ट कोडोन इज AUG. यस, थैंक्स लॉर्ड. अब चलते हैं आगे. अगला टॉपिक है टॉपिक नंबर 38. सही हमने 35 किया, 36 किया, 37 किया, अब 38 टॉपिक है ट्रांसक्रिप्शन 2. इसका क्विज ले लेते हैं. So in eukaryotes, in order to initiate transcription, यूकैरियोट्स में ट्रांसक्रिप्शन को इनिशिएट करने के लिए promoter sequence must be present. None of these RNA strand must be present. RNA polymerase must be present. तो ये promoter sequence must be present. ठीक है. उसके बाद फिर ये हो गया करेक्ट. चलते हैं आगे. थैंक्स लॉर्ड. अब चलते अपनी वीडियो को इसको अच्छे से सुन लेते हैं. ठीक है.
Dear students, in this module, I'm going to talk about transcription. Transcription is a very important process by which information from the DNA is converted into the RNAs and onwards for protein synthesis. As you know, these proteins formed as a result of this process are very useful in the formation of cells as well as in the cellular life. As I just mentioned, cells are built up of proteins and carbohydrates, and the DNA inside the nucleus is responsible for the creation of newer cells. In order to form a new cell, the information within the DNA has to be conveyed or converted into proteins. Let's take a look at how this process works step by step. In this figure, you can see that the DNA is coding for an RNA. This process is followed by encoding of the RNA into the protein. So this entire process is a part of what we call the central dogma. The central dogma can then be broken into two components. The first part being the relaying of information from the DNA to the RNA, and the second part being the transfer of this information from the RNA to the proteins. In this module, we're going to focus on the first part, which is called transcription. This process, to define, is responsible for converting the DNA bases into the RNA bases. The DNA is double-stranded while the RNA is single-stranded. Following this, the process of translation is there. We will look at it later. During this transcription, information from the DNA is converted into the RNA. A copy is therefore created, which is used in creating the proteins, which are the executive molecules or molecules responsible for execution of the information. So transcription, therefore, is a process by which the 4 nucleotide bases within the DNA are converted into the RNA. A DNA molecule may have hundreds or thousands of these bases, and therefore the RNA molecule that is produced will have a similar number of bases in it as well. However, there are two differences that you need to remember. The first one is that in an RNA molecule, there is no thymine, and thymine is replaced by uracil. Secondly, the RNA molecule is a single-stranded molecule instead of the DNA, which was the double-stranded version. Moreover, A only couples with the T in the DNA and G only couples with the C. However, in the RNA, A gets coupled with a U and C stays coupled with the G. So here is the process in detail. As you can see, the double-stranded DNA is shown here, and the nucleotide bases T and A are coupled. Similarly, nucleotide base C is coupled with G, and so on and so forth. Typically, G and C never couple with T and A, and vice versa as well. So the process of transcription then is the process which decodes information from the DNA and prepares a copy of this information towards preparing an RNA molecule. The DNA to RNA conversion, as I just mentioned, involves two important steps. One, that the double-stranded DNA is converted into a single-stranded RNA, while thymine is replaced by uracil. The RNA then is constituted by 4 bases as well, and an RNA molecule may have multiple copies of these 4 nucleotide bases. As I just mentioned, they include A, U, G, and C. So in conclusion, DNA gives rise to an RNA. The DNA had 4 different types of bases, A, T, C, and G, while the RNA has one difference from that is uracil replaces thymine. More so, the DNA is double-stranded, while the RNA is single-stranded. These RNA molecules in turn later undergo a process called translation to produce proteins.
सो फ्रेंड्स, हमारी वीडियो हो गई है कंप्लीट. हम चलते हैं अपनी रीडिंग की तरफ. इसको कर लेते हैं डाउनलोड. All cells are made up of proteins, carbohydrates and lipids, and for these cells DNA codes the information which makes the RNA protein both. ठीक है, RNA and protein both. सही है. तो यहाँ पर आगे ट्रांसक्रिप्शन हो रही है DNA से RNA में, जो कन्वर्शन हो रही है, डिकोडिंग हो रही है, तो उसे कहते हैं ट्रांसक्रिप्शन. उसके बाद ट्रांसलेशन हो रही है, किसमें होगी, RNA से जो प्रोटीन बनेगी उसमें, उस प्रोसेस करेंगे ट्रांसलेशन. और सारा का सारा क्या है, सेंट्रल डॉग्मा. और जहाँ पर इंफॉर्मेशन है जो DNA में, वो कॉपी हो रही है इन्फॉर्मेशन पर. उसके बाद जो और प्रोटीन बनती है वो होती है एग्जीक्यूशन ऑफ इन्फॉर्मेशन. ठीक है. उसके बाद हमारे पास ट्रांसक्रिप्शन इज द प्रोसेस बाय विच इंफॉर्मेशन इन अ स्ट्रैंड ऑफ DNA इज कॉपीड इनटू अ न्यू मॉलिक्यूल ऑफ मैसेंजर RNA. DNA safely and stably stores genetic material in the nuclei of cells as a reference or template. Meanwhile, mRNA is comparable to a copy from a reference book because it carries the same information as DNA but is not used for long-term storage and can freely exit the nucleus. Although the mRNA contains the same information, it is not an identical copy of the DNA segment because its sequence is complementary to the DNA template. Transcription is carried out by an enzyme called RNA polymerase and a number of accessory proteins called transcription factors. Transcription factors can bind to specific DNA sequences called enhancer and promoter sequences in order to recruit RNA polymerase to an appropriate transcription site. ठीक है. Together the transcription factors and RNA polymerase form a complex called the transcription initiation complex. This complex initiates transcription and the RNA polymerase begins mRNA synthesis by matching complementary bases to the original DNA strand. The mRNA molecule is elongated and once the strand is completely synthesized, transcription is terminated. The newly formed mRNA copies of the gene then serve as blueprints for protein synthesis during the process of translation. सही है. उसके बाद में हमारे पास mechanism. The above mechanism explains the process of transcription in a very simple way. DNA codes the information and converted into RNA, where mRNA copies the information and it executes the information in cell, and amino acid combines with each other according to coded information of DNA and protein formation takes place. This is known as translation. Okay, next is RNA versus DNA. Molecule of DNA contains only 4 base pairs: adenine, thymine, cytosine and guanine, which are repeated thousands of times, and adenine pairs with thymine by 2 hydrogen bonding, while cytosine binds with guanine by 3 hydrogen bonds. ठीक है, so same like DNA, the RNA contains 4 base pairs, but thymine is replaced by uracil and RNA is single stranded. ठीक हो गया. कंक्लूजन में हमारे पास में आ गया. Conclusion में अगर DNA has 4 bases: adenine, cytosine, guanine, thymine and RNA. And RNA also has 4 bases: adenine, guanine, cytosine and uracil. DNA is double stranded whereas RNA is single stranded. DNA just codes information from proteins but RNA helps in making proteins. ठीक हो गया. उसके बाद हमारे पास आ गया, हमारे पास हमारी PPTs आ गई हैं, उसको भी रीड कर लेते हैं. ट्रांसक्रिप्शन के अंदर आ गया बैकग्राउंड. सेल्स आर बिल्ट अप ऑफ प्रोटीन्स एंड कार्बोहाइड्रेट्स. दीज मॉलिक्यूल्स आर प्रोड्यूस्ड आफ्टर ट्रांसफॉर्मेशन ऑफ DNA into proteins. What are the salient steps underlining this transformation? So this is central dogma: transformation of information from DNA to RNA. This is the copy of information. This is known as transcription, and after that RNA makes up proteins by the process of translation, and this is known as execution of information copied from DNA. ठीक है, and then it helps to make cells. ठीक है, उसके बाद फिर transcription. DNA is constituted by 4 bases. These are adenine, cytosine, thymine and guanine. DNA molecule may contain thousands of adenine, cytosine, thymine and guanine. Adenine pairs with thymine and cytosine pairs with guanine. ठीक है, इसके बाद फिर ये सारे 13 की बेस है. इसके बाद फिर transcription, process of transcription decodes these bases in the DNA based on the information received after decoding DNA and RNA molecule is encoded. उसके बाद हमारे पास आ गया ट्रांसक्रिप्शन. उसके बाद में ट्रांसक्रिप्शन में है RNA is consisted by 4 bases. These are adenine, cytosine, uracil and guanine. And RNA molecule may contain thousands of adenine, cytosine, uracil and guanine. And adenine pairs with uracil, only cytosine paired with guanine. ठीक है. Conclusion में आकर DNA gives rise to RNA. DNA has adenine, thymine, cytosine, guanine, while RNA has adenine, uracil, guanine and cytosine. ठीक है. DNA is double stranded while RNA is single stranded. RNA helps produce proteins. ओके जी, हमारा बहुत अच्छे से समझ आ रहा है सच तो ये टॉपिक. ठीक है, आप लोग भी आपको पा रहे होंगे समझ में. अब देखते हैं अपनी पोस्ट असेसमेंट की तरफ. इसका भी ले लेते हैं. ठीक है. इन यूकैरियोट्स इन ऑर्डर टू इनिशिएट ट्रांसक्रिप्शन वो होना चाहिए प्रोमोटर सीक्वेंस. हाँ, प्रोमोटर सीक्वेंस प्रेजेंट होना चाहिए. इसके बाद में researchers first identified parts of genes that are spliced out of mRNA and are not included in the final protein product. What are these regions? Introns, intergenetic regions, micro RNA exons. हाँ, मेरे ख्याल में ये है exons I guess. ओ इंट्रॉन्स होंगे फिर वो. ठीक है, चलो इसको कर लेते हैं क्रॉस. अब चलते हैं, हमारे पास 38 टॉपिक्स हो गया. अब हम चलें अपनी 39 टॉपिक की तरफ. ठीक है. टाइप्स ऑफ राइबोन्यूक्लियोटाइड्स. सही है, इसका क्विज ले लेते सबसे पहले. सही है, इसका क्विज हमने अब यहाँ पर ले लेना है. तो फॉलोइंग कोडोन इज mRNA स्टार्ट कोडोन दैट इनिशिएट्स ट्रांसलेशन. AUG है ये तो स्टार्ट कोडोन. आगे चलते हैं अपनी वीडियो की तरफ. वीडियो को अच्छे से सुन लेते हैं.
There are 4 main types of ribonucleotides present in the cells, and it depends upon the types of nitrogenous bases present in a ribonucleotide. You know the structure of a ribonucleotide, right? There are 3 components. One is pentose sugar, ribose sugar. The other one is phosphoric acid, and the third one is nitrogenous base. the two components, that is phosphoric acid and a ribose sugar, they are same in all the ribonucleotides, and the only difference is in the nitrogenous base. So as you know, there are 4 types of nitrogenous bases present in a ribonucleotide. One nitrogenous base is present in one ribonucleotide, but overall there are 4 types of ribonucleotides. So there are 4 types of nucleotides which are present in a ribonucleic acid. These 4 types, they depend on the type of nitrogenous base. As you can see, this colored portion is a nucleoside. You know, this is a nucleoside because a pentose sugar and a nitrogenous base, when they both combine, they make a nucleoside. And here the nitrogenous base is adenine, so this nucleoside is adenosine. And when to this adenosine a phosphoric acid is attached, it becomes a nucleotide. So this adenosine becomes adenylate. Let's move to the second type of ribonucleotide present in the nucleic acids. Here it is guanine. So in this colored portion, a guanine joins with a pentose sugar and they make a guanosine. And when to this guanosine a phosphoric acid is attached, it make a guanylate. So next, here are 2 other types of ribonucleotides. Again, the colored portion, this colored portion is a uridine because the nitrogenous base present here, this is uracil. So to this uridine, one phosphoric acid group is attached and it becomes uridylate. This is 3rd type of ribonucleotide present in the nucleic acids. The 4th and the last type of ribonucleotide is cytidylate. In a cytidylate, the nitrogenous base is cytosine and this pentose sugar and this phosphoric acid is present. So when a cytosine attaches with pentose sugar, it makes cytidine. And when to this cytidine a phosphoric acid is attached, a complete nucleotide, that is cytidylate, is formed. So these 4 types of ribonucleotides are present in the ribonucleic acids. Next, how these ribonucleotides are joined together. Because I said ribonucleic acid is a polymeric compound. So many ribonucleotides join together to make a molecule. So here you can see this is one ribonucleotide and this is second ribonucleotide. So in this ribonucleotide, one phosphoric acid is attached to its 5 carbon, and this attachment is through ester linkage. So the hydroxyl group present here in this phosphoric acid, it attacks to the hydroxyl group present on the pentose of next ribonucleotide. So in this way, a condensation reaction takes place here. A water molecule is removed, and here you can see these 2 ribonucleotides, they join together and they make a dinucleotide, in which these 2 nucleotides are linked together through phosphodiester linkage. Why this is called phosphodiester linkage? Because here one phosphate group is present, and this phosphate group, it is linked with one pentose with a ester linkage, and the second ester linkage is present here. So all this is one phosphate and 2 esters, so it is called phosphodiester. So in this way, a polymeric compound consisting of many ribonucleotides are formed. So in this way, many ribonucleotides they join together and they make a polymeric chain, which is called a polyribonucleotide or a ribonucleic acid.
सो फ्रेंड्स, फाइनली हमारी वीडियो होगी है कंप्लीट। हम चलते हैं अपनी रीडिंग की तरफ। रीडिंग कर लेते हैं। सही है, इसको बस साथ में डाउनलोड कर लेते हैं। और यहाँ पर है देयर आर मेनली 4 टाइप्स ऑफ ribonucleotide. देयर आर मेनली 4 टाइप्स ऑफ ribonucleotide डिपेंडिंग अपॉन द टाइप्स ऑफ नाइट्रोजन बेसिस प्रेजेंट इन आरएनए। ठीक है, क्या है एडिनिलेट, जिसमें होता है एडिनोसाइन 5' और मोनोफॉस्फेट। ठीक है, और उसके बाद ग्वानिलेट, जिसमें होता है गोनासाइन 5' और मोनोफॉस्फेट। ठीक है, उसके बाद यूरिडिलेट, जिसमें होता है यूरिडाइन 5' मोनोफॉस्फेट। ठीक है, साइटिडिलेट, जिसमें तो साइटिडाइन 5' मोनोफॉस्फेट। सही हो गया। उसके बाद हाउ टू राइबोन्यूक्लियोटाइड जॉइन। अब जॉइन कैसे करते हैं? फॉस्फोडाइस्टर लिंकेज से जॉइन करते हैं। यहाँ पर कंडेंसेशन हो जाती है और ये जो है वो इसके 5 कार्बन के साथ जुड़ा हुआ है शुगर के। ठीक है, उसके बाद फिर ये फॉस्फेट ग्रुप जो है ये कंडेंसेशन कर लेता है दूसरे वाले शुगर के और हाइड्रोक्सिल ग्रुप पर अटैक करता है और उसके बाद कंडेंसेशन हो जाती है, वो एक वाटर मॉलिक्यूल रिमूव हो जाता है और यहाँ पर बन जाता है फॉस्फोडाइस्टर। एक ये ईस्टर बोन और एक ये ईस्टर बोन। फॉस्फोडाइस्टर 1 फॉस्फेट 2 ईस्टर। ठीक है, अभी सर ने हमें समझाया। ऐसे ये प्लस वाटर फॉर्मेशन होता है। ठीक है, राइबोन्यूक्लियोटाइड फॉस्फेट शुगर 5' 3' बेस। ठीक है, तो ये तो हमें पता है। सही है, चलते हैं हम आगे। अपनी पिपरीज को रीड आउट कर लेंगे आप फ़ौरन। 1 सेकंड से पहले 1 सेकंड का टाइम ज़ाया करने से पहले हम अपनी पिपरीज को निकालेंगे और रीड आउट करेंगे। टाइप्स ऑफ राइबोन्यूक्लियोटाइड्स, देयर आर मेनली 4 टाइप्स ऑफ राइबोन्यूक्लियोटाइड डिपेंडिंग अपॉन द टाइप्स ऑफ नाइट्रोजन बेसिस प्रेजेंट इन आरएनए। सो ये हमने अभी पढ़ा है, ये भी पढ़ा, ये पढ़ लिया हमने, ये पढ़ लिया। सो चलो जी, हमने इसको इस सारे स्ट्रक्चर्स को देखा भी है, सर ने वीडियो में अच्छे से समझा दिया। ठीक है, तो अब चलते हैं आगे। आगे हमारे पास में हमारी पोस्ट असेसमेंट है, इसको ले लेते हैं। बिस्मिल्लाहिर्रहमानिर्रहीम, लेट्स गो। टाइप्स ऑफ आरएनए दैट कैरीज जेनेटिक इंफॉर्मेशन फ्रॉम डीएनए टू द प्रोटीन बायोसिंथेटिक मशीनरी ऑफ द राइबोसोम्स। ठीक है, तो ये है ट्रांसफ़र आरएनए। ठीक, उसके बाद व्हिच ऑफ द फॉलोइंग आरएनए स्ट्रक्चर इज सिमिलर टू क्लोवर लीफ। ये क्लोवर लीफ ट्रांसफ़र आरएनए का स्ट्रक्चर। अच्छा, तो इसको क्लोवर लीफ से अच्छा, तो वो mRNA हो सकता है। ठीक है, इसको कर लेते हैं क्रॉस। उसके बाद यहाँ पर हमारे पास अगला टॉपिक आ जाता है, टॉपिक नंबर 40, टाइप्स ऑफ आरएनए। ठीक है, टाइप्स ऑफ आरएनए आ गया, इसको टेक क्विज कर लेंगे सबसे पहले तो। ठीक है, टाइप्स ऑफ आरएनए दैट कैरीज जेनेटिक इंफॉर्मेशन फ्रॉम डीएनए टू द प्रोटीन बायोसिंथेटिक मशीनरी ऑफ द राइबोसोम्स। ये है टीआरएनए। इसको कर लेते हैं सेव। जेनेटिक इंफॉर्मेशन फ्रॉम डीएनए टू द प्रोटीन बायोसिंथेटिक मशीनरी ऑफ राइबोसोम्स। टीआरएनए होना तो चाहिए था, पता नहीं क्यों। द ट्रांसक्रिप्शन फैक्टर्स असिस्ट द आरएनए polymerase in locating active site stop codon mutation promoter. Transcription factor assists the RNA polymerase in locating promoter को locate करने में I think I guess yes promoter, it's true। अब चलते हैं अपनी वीडियो की तरफ से, अच्छे से सुन लेते हैं।
Let us see different types of RNAs. As you know, only one type of DNA is present in the cells, but the ribonucleic acids, they are of 3 types present in the living organism, and these are messenger RNA, or generally called as mRNA, transfer RNA or tRNA, and ribosomal RNA or rRNA. So these 3 types of ribonucleic acids are present in the cells of living organisms. first type is the messenger RNA. As you know, the process of protein synthesis is carried out in the cytoplasm, and you also know the DNA is only present in the nucleus. It doesn't go out to the cytoplasm. So how it conveys message from nucleus to the site of protein synthesis in the cytoplasm to synthesize the protein? So definitely there will be some messenger, and this messenger is a ribonucleic acid, and this messenger is mRNA or messenger RNA. It is that type of RNA that carries genetic information from DNA to the protein biosynthetic machinery, that is ribosomes, which is present in the cytoplasm. This mRNA provides the templates that specify amino acid sequences in the polypeptide chain, because this mRNA molecule, it takes the information from DNA, so it contains the templates copied from DNA, and these templates are then translated in the form of sequence of amino acids in a protein molecule. So this mRNA is synthesized in the nucleus from DNA, and then after synthesis it is transported from nucleus to the cytoplasm. The process of synthesis of mRNA on a DNA template in the nucleus is known as transcription. This mRNA molecule, as you know, it is for the synthesis of a particular protein, and the synthesis of a protein is specified by a particular gene. So actually this mRNA molecule contains information of a particular gene. It may contain the information of one gene or more than one gene. So it may be monocistronic or polycistronic. In case of monocistronic, this mRNA molecule, it is only coding one gene, that is, it is responsible for the synthesis of only one type of protein. While in case of a polycistronic mRNA, it contains the information for more than one genes, so it is responsible for the synthesis of more than one types of the proteins. So the length of mRNA molecule, it is variable, because it may be a monocistronic containing one gene, or it may containing many genes, and then the length of gene is also very variable. So the length of mRNA molecule is also variable. The second type of ribonucleotide which is present in the cytoplasm, that is transfer RNA or tRNA. Transfer RNA serves as an adapter molecule in the process of protein synthesis. How it acts as adapter? To one end, this tRNA molecule is linked with an amino acid. So it carries that amino acid to the site of protein synthesis, that is to the ribosome. So in this way it is acting as an adapter. So the main function of transfer RNA, as the name is indicating, is the transfer of amino acid from different sites of the cytoplasm to the site of protein synthesis in the cytoplasm. These transfer RNA molecules, they pair with mRNA in such a way that the amino acids which are carried by these tRNA molecules, they are joined to the growing polypeptide chain in a correct sequence. So as you can see in this illustration, this is tRNA molecule, and it contains an amino acid which is covalently attached with this tRNA molecule, and here you can see a specific sequence of 3 letters. This sequence is called anticodon, and this anticodon is complementary with the sequence of 3 letters on mRNA. So this tRNA molecule will combine with the mRNA with the help of this specific anticodon, and when this tRNA molecule attaches here with this protein synthetic machinery, it hands over its amino acid to the already growing polypeptide chain, and after handing over its amino acid, it goes out of the synthetic complex, and here you can see this tRNA molecule is without any amino acid. So in this way, new tRNAs with new amino acids come, they give their amino acid to the polypeptide chain, and they go out without the amino acid. In this way, they help in the synthesis of proteins. The third type of ribonucleic acid is ribosomal RNA or rRNA. Ribosomal RNAs are components of the ribosomes, and ribosomes are the protein synthetic machineries. Ribosomal RNA is a predominant material in the ribosome, and it comprises about 60% of ribosomes by weight. It has a number of functions to perform in the ribosome during the process of protein synthesis.
सो फ्रेंड्स, फाइनली हमारी वीडियो हो गई है कंप्लीट. हम चलते हैं अपनी रीडिंग की तरफ. चलो जी, फटाफट से अपने रीडिंग के पास आ जाते हैं. PPTs को कर लेते हैं डाउनलोड. देयर आर मेनली 3 टाइप्स ऑफ ribonucleic acids RNAs present in the cells of living organisms: messenger RNA, mRNA, transfer RNA, tRNA, ribosomal RNA, rRNA. And we have here हमारे पास है messenger RNA, mRNA. It is a type of RNA that carries genetic information from DNA to the protein by synthetic machinery of the ribosomes. It provides the templates that specify amino acid sequences in polypeptide chains. The process of the process of forming mRNA on a DNA template is known as transcription. It may be monocystronic or polycystronic. Monocystronic or polycystronic. The length of mRNA molecules is variable and it depends on the length of gene. ठीक है. उसके बाद transfer RNA serve as adapter molecules in the process of protein synthesis. They are covalently linked to an amino acid at one end. They pair with the mRNA in such a way that amino acids are joined to a growing polypeptide in the correct sequence. ठीक है. और ये वाला हो गया इसका सीक्वेंस. एक tRNA आ रहा है अमीनो एसिड के साथ में, और ये जो इंफॉर्मेशन दे देता है इसको राइबोसोम के rRNA को. उसके बाद फिर वहाँ से और फिर उसके बाद यहाँ पर प्रोटीन की सिंथेसिस हो जाती है और फिर ये खाली हाथ वापस चला जाता है. ठीक है. उसके बाद यहाँ पर आ जाते हैं राइबोसोमल RNA. राइबोसोमल RNA is the third type. Ribosomal RNAs are components of ribosomes. RNAs are predominant material in the ribosomes, constituting about 60% of its weight. It has a number of functions to perform in the ribosomes. ठीक है. उसके बाद हमारी PPTs आ जाएंगी. चलो जी, अब हम PPTs को अच्छे से स्टडी कर लेते हैं. Types of RNAs. There are mainly 3 types of ribonucleic acids. RNAs present in the cells of living organisms: messenger RNA, mRNA, transfer RNA, tRNA, ribosomal RNA. राइबोसोमल आरएनए आ गया। यहाँ पर टाइप्स ऑफ आरएनए में राइबोसोमल आरएनए, आरआरएनए, राइबोसोमल आरएनएज आर कंपोनेंट्स ऑफ राइबोसोम्स। आरआरएनए इज a predominant material in the ribosomes, consisting about 60% of its weight. It has a number of functions to perform in the ribosomes, of course, as it is making proteins directly. ठीक है, उसके बाद हमारे पास में आ जाता है पोस्ट असेसमेंट। पोस्ट असेसमेंट में हम लोग अपना क्वेश्चन ले लेते हैं। tRNA are linked together, linked to an amino acid at one end through covalent bond. Dash performs number of functions in the ribosomes: messenger RNA, micro RNAs, ribosomal RNA. So both are correct. Thanks Lord. इसको कर लेते हैं क्रॉस। अब इस वीडियो में इतने ही टॉपिक्स करेंगे हम, अगले 41 से 45 तक इंशाल्लाह करेंगे अगली वीडियो में।